Assalamualaikum and a very good day everyone. We are Group 1 from Kuliah 4 are going to talk about asexual reproductive strategies, specifically in plants. So we know that plant can reproduce sexually, right? And the organ of sexual reproduction is the flower itself, which have the male and female gametes. By looking at this sexual reproduction process, you might be wondering, can plant reproduce asexually though? Well, the answer is yes. Plant can reproduce asexually without the involvement of male and female gametes. But how? So that's why in this video, we are going to tackle about the asexual reproduction strategies in plants, but primarily focus on tissue culture.
Before we start, here is some facts about asexual reproduction in plants. What is asexual reproduction? Asexual reproduction is a method that produces a plant that are genetically identical to the parent plant because no mixing of male and female gametes takes place. Because of their offspring is genetically identical to the parent plant, it is called a clone. Traditionally, these plants survive well under stable environment conditions when compared with plants that are produced from sexual reproduction because they carry genes identical to those of their parents. There are two parts of plants that can be used for asexual reproduction, which are stolons and rhizomes. What are stolons? Stolons are stems which grow at the soil surface or just below ground that form adventitious roots at the nodes and new plants from the buds. As you know, the nodes of stems are regions where new growth can occur. In the case of stolons, a new shoot system appears above the node and a new root system appears below the node. So here are some of example plants that spread with stolons, which are strawberries and mint. And as we can see, the larger plant on the left is the parent plant and the smaller plant on the right is the asexual clone that has arisen at a node. What is rhizome? Technically, a rhizome is a stem that grows underground. It usually grows horizontally just below the soil surface. Since it is a stem, it has nodes and is able to put out other stems, usually straight up and above ground. This means a patch of what looks like several individual plants grouped near each other may actually all be shoots of the same plant put up by the same rhizome. So here are some of example rhizome plants that are used directly in cooking, such as ginger, fingerroot, and lotus.
Now that you know about asexual reproduction strategies, let's get back to our highlighted topic, which is tissue culture. So what does it mean by tissue culture? Plant tissue culture is one of the techniques to grow plant cells, tissue or organs under sterile conditions on a nutrient culture medium of non-composition. It is widely used to produce clones of a plant in a method known as micropropagation. Techniques for tissue culture vary, but most begin with cells from the meristem of the parent plant. Merismatic cells are actively dividing cells are grown on agar medium in tube, flasks, or petri dishes. The medium contains auxin, cytokinins, water, and various nutrients. At first, a mass of unspecialized cells called coelus is formed. This combination of auxin and cytokinins will signal organogenesis or organ formation and initiate the formation of roots and shoots until a fully developed plantlet is formed. Plantlets then can be transferred in their sterile containers to grow for transplantation in a greenhouse or field.
Now you already know the techniques of tissue culture, so I'm going to explain a few benefits of plant tissue culture. The first benefit is the new plantlets from tissue culture techniques can be grown in a short amount of time. Second, the new plantlets and plants are also more likely to be free of virus and disease. Third, only a small amount of initial plant tissue is required in the process. And the last is, the process is not dependent on the seasons and it can be done throughout the year. Okay, now I'm going to explain what is cell suspension culture. Cell suspension culture is a type of culture in which single cells or small aggregates of cells multiply while suspended in agitated liquid medium. The suspension culture of single cells helps in the understanding of growth and developmental process of the plant. In conclusion, we know that all plants can reproduce sexually or asexually, even though asexual reproduction does not contribute into evolution as it does not cause the variation, but still it is important for the growth of the organism. Meanwhile, for the plant tissue culture, it is important for a researcher to be ethical while performing the tissue culture, as this technique comes great responsibility to humans. The plant tissue culture also represents the most promising areas of application and giving an outlook for the future. And one thing that we need to remember is the plant tissue culture is only our hope to end the world hunger. That is all from us. Thank you.
Assalamualaikum and a very good morning to everyone. So we are here today with Kak Fatin Aini, a fresh graduate Bachelor of Science Agrotechnology with Honours Agronomy from UiTM. Okay, so in this interview, we will unveil a bit about tissue culture. So Kak Fatin, the first question is, what are the benefits of tissue culture in plants and why was this method introduced?
The benefits are the new plants can be grown in a short amount of time. We only require a small amount of initial plant tissue, and the process is not dependent on the season and can be done throughout the years. And finally, help to supply the consumer market with new subspecies and variety. The purpose of doing tissue culture are to produce a mass multiplication of plants in a short time and to produce plants that have high resistance to disease.
Next question. Can we do tissue culture on all plants? If not, what is the example of plants that is commonly used in tissue culture?
Okay, thank you, Nuri. Okay, in my opinions, we can do tissue culture on all plants, but the things that make them differ are the time period for them to grow during the initial phase. For example, oil palms need 3 to 5 years for the callus to grow, and the common plants that are used for tissue culture are banana.
Can you please explain briefly how tissue culture is conducted in the lab?
We can do tissue culture in lab by cutting or scrap a tissue sample from the parent plants, and the tissue samples are then placed in agar grow mediums that contain auxins and nutrients. The sample will develop into a tiny plants, and then we transplant them into a compost.
Okay. The next question is, what is the most crucial thing that we should be aware of when doing tissue culture?
The crucial things that we should be aware when doing tissue culture are the contamination rates and the tissue culture technique. The aseptic technique need to be improved so that the contamination rate lower, and tissue culture technique need to improve so that to improve the growth of the explant.
Our last question is, if you have the chance to promote tissue culture to the public, what would you tell them?
Okay. Tissue culturing is an important advancement in biological research that allow investigators to analyze the growth, development, and function of the cells. It has both advantages and disadvantages, so it is in our hands to use it carefully.